Okay, hello everyone. My name is Matt Selensky. I'm a fifth-year PhD candidate in Maggie Osborne's group, and I study cave microbial ecology in my research. And so one of the things that we do as microbial ecologists all the time is try to study complex microbial communities without actually trying to grow them, because most of them can't be grown with traditional methods that microbiologists try to use. And so long story short, this is a horrendous oversimplification of what we do, but we essentially extract DNA, amplify taxonomic marker genes, and then do some data processing to essentially get a table of counts of like relative abundances of each species in each sample. And oftentimes what we do in our lab is we try to compare that to a bunch of other different types of data to try to understand what these microbes might be doing. And so you can easily get up to like 10,000 different unique species in a data set, which becomes a really like just computationally challenging problem, as well as wrapping your head around what's happening in the ecosystem. And what a lot of people do is kind of do a correlational type analysis with pairwise relationships, where you look at the relative abundance of one microbe versus another, you look at the direction of its relationship, the strength of that relationship, and you can also do the same thing with environmental variables. And network analysis is a really good way to kind of bring all of that in together. And it's been a tool that's been used by microbial ecologists more and more frequently. But what I've noticed about a lot of social kind of network analysis tools that people use in the field is that they weight both of these pairwise relationships the same. If you notice here, microbe 1 and microbe 2 only has 2 observations, and it has a really beautiful, perfectly correlated line here, but there's 2 observations. That's not a very useful line in my opinion. And it weights it the same as something like this, where you have more observations. And so what my software does, which is available at this link here, is it basically looks for these pairwise relationships that are robust enough to be included in a network, where you can look at these sorts of variables to try to visualize what that ecosystem structure is like. And so putting it in with some real data from the Yucatan aquifer that I'm collaborating with Trish Beddoes on, this is what one of the outputs will look like. And what the software also does is it employs an algorithm called EBC, or edge betweenness clustering, to try to kind of parse out what the subnetwork structures are. And so each individual node here is an individual microbial species, if it's a circle, or an environmental variable, if it's a square. And what I'm trying to do is understand how all these different individual players are interacting with each other through this analysis. And another thing that it does is looks at the abundance of these things here throughout the data set. And I'll just end it here by saying you can apply it to the data and you can see biogeographic patterns in the data set. And with that, I was told to stop, please. Questions. Yeah.
Sorry. I was gonna give students a chance. Can you give so far away from microbiology? Can you give an example of something you were able to discover with this network?
Yeah, so if you could go back to the other slide. So one thing that really popped out here is, so if you see sort of how these samples first are clustered by how the communities are similar to each other. So where the bars are, like that's one sample and it's more similar to the samples next to it. And what we found here was this big, this big kind of light purple one here is mostly found in a region of the aquifer that's an open pit cenote that has a lot of different, it's kind of distinct geochemistry, like it has a lot of sulfur. And as a result, all the microbes that make up this cluster, could you hit one more, actually pop out here where there's this density interface in the aquifer where that sulfur accumulates, organic matter accumulates, and we're seeing those microbes pop out there. So it was a good way to kind of like orient myself to be like, okay, well, we see these sulfur oxidizing microbes in this cluster here. There are other sulfur oxidizing microbes in the aquifer, but they're different than the ones that we see in this really distinct environment. So that was kind of fun.
Thank you.
Yeah.
With that.
Thank you.